All right. So welcome to my grand rounds. Chose a topic that I thought was kind of boring actually when I was thinking about it a few weeks
That's why
ago.
you decided to do
Like,
it.
as I got into it, I decided that I didn't actually think it was boring, but I thought it was remarkably frustrating, especially in transplant nephrology. So it's more frustration than boredom. So let's get into it. BK, polyomavirus, and disease. No disclosures to report. I have three main objectives. Number one, I'd like to describe a basic understanding of the virology of the BK virus, including major capsid proteins and genome transcription. Explain the natural history of BK virus disease in the current transplant era. That's important. Review the current guidelines on BK virus management in the kidney transplant recipients and the predominant evidence supporting those guidelines. Okay, get this out of the way here. So in lieu of a patient presentation, as we commonly do, I chose to do something a little bit different. I thought I would be good to take kind of a historical perspective. I like to do timelines to kind of to frame what we're going to talk about. I want to highlight here some of the major events in kidney transplantation. And then we'll kind of relate that back to how we understand the natural history of BK virus in the modern transplant era. So number one, 1971 is when BK was isolated from a kidney transplant recipient who had ureteral stenosis and was observed to have urinary inclusion-bearing cells. 1983 was when cyclosporin gained FDA approval. 1993 was when the first definitive biopsy-proven case of BK virus-associated nephropathy was described. It was in 97 that tacrolimus was FDA approved for use in kidney transplant recipients. 1997 was the first Banff consensus guidelines for renal allograft pathology. So from the mid-1990s to the early 2000s, I've seen it described in a few cases, a few reports that BKVAN frequently resulted in graft loss with rates as high as 50 to 100%. So that prompted in 2003 the first polyomavirus-associated nephropathy international conference. Out of that conference in 2005 came the first international consensus guidelines on the management of polyomavirus nephropathy. So between 2005 and the present, with the increased recognition of BK virus disease and nuanced management among transplant centers, we saw a reduction in graft loss due to PVAN. 2018 came the Banff update, where the BK grading system came into force. In 2019, we had the AST guidelines for solid organ transplantation. And then just last fall, the International Society of Transplantation published its second international consensus guidelines. So we're going to talk about BK, right? It's this parasitic virus. It has lytic replication in our cells. It causes these things we're all familiar with: BK virus-associated nephropathy, graft loss, hemorrhagic cystitis, ureteral stricture, and increasingly it seems to be associated with urothelial bladder cancer. So what is BK? A little bit of virology. We're going to get a little deep here. It's a member of the Polyomaviridae family of double-stranded DNA viruses. It's one of 14 polyomaviruses with a human host. It's small, non-enveloped viral genome, and it's surrounded by viral capsid proteins. JC virus and Merkel cell virus, of course, are in this family. What does it look like? Quite interesting, actually. So we have here, this is an early model of polyomavirus built from crystallography, that it shows this very beautiful icosahedral shape. The viral capsid, that consists of 360 repeating copies of the major capsid protein, VP1, and that's the color coding scheme there. So they in turn form 72 pentons to form the icosahedral structural lattice. And up until relatively recently, and in scientific terms, this largely were our understanding of the structure of the polyoma virus family. So this is really cool. So it's quite nice that you can grow polyomavirus in cell culture. So what we see here is electron microscopy of the polyomavirus in renal tubular epithelial cells. If you look real closely, you can kind of see that icosahedral shape. But on the left-hand side of the screen, this is really cool. When I was going through the literature on this, there's this group of researchers out of the University of Leeds in the UK, and kind of banking on the fact that you can culture BK virus in cell cultures. What they've done is they grow the virus. Okay, they then harvest the virus. They place the virus on this carbon matrix. They freeze it. And then they take it to a cryo-electron microscope, place it under the microscope, peer down the microscope, and they're able to generate these images of the BK virus in really exquisite detail. And you can kind of see the individual structures there. But not only that, they can get to even more detail, keeping your mind on those individual structures. They're able to blow it up. So you can see this, this VP1 protein, that a whole conjunction of them, 7 of them, from the outside of the virus, and then they can flip it 180 degrees and you can see it from the inside. I have a lock and key here because this has fantastic implications for pharmaceutical development, right? Specifically targeted pharmaceuticals against the BK virus. Keeping on that theme of the lock and key, so what about the cell surface, the tubular epithelial cell surface? What's the receptor that the BK virus binds to? We think we know. We think we have a good idea. That's the B series ganglioside. So this group of researchers, they know this. So they'll grow the virus in culture, and then they'll harvest the virus, and they will bathe that virus in this molecule, freeze that, take it back to the microscope, and see what they got. And they get this right here, right? So this is the BK virus, and that is, in purple, that is the B series ganglioside affixed to it. Really quite fascinating and offers a lot of potential for therapeutics. So now let's look inside, right? Like this guy's doing up there at the top. What's inside the viral genome? So there are 4 major genotypes of the BK virus, and that's based on sequence heterogeneity of the VP1 protein. Subtypes 1, 2, 3, and 4. Subtype 1 is the most common. It's found worldwide. That's usually the one that causes disease. Subtype 4 has pockets in Southeast Asia and Europe. So I had to ask myself the question, well, what if I had BK virus and I have subtype 4? Oh, but maybe my lab is testing for subtype 1 and we won't catch it. That's okay. Most clinically validated BK virus DNA PCR assays are designed to target conserved regions of the BK virus genome, whether it be a conserved region of VP1 or the non-coding control region. Well, we'll go through that. All right, so what's packed inside? The genome, it's about 5.1 5100 base pairs. DNA genome, it's circular, it's packed around host histone proteins, and it mainly has 3 coding regions: the early coding region, the regulatory region, or the non-coding control region, and the late coding region. So the early coding region, this codes for the large and small tumor antigens. And that's interesting because that's actually what the SV40 stain targets when we do the pathology. And so these proteins that are expressed, they're expressed prior to genome replication. And then the tumor antigen, that has a propensity to bind P53 and the retinoblastoma family. And so that results in commencement of the cell cycle in hosts and then subsequent persistent infection. The non-coding control region, or the regulatory region, that contains the origin of viral DNA replication, as well as transcriptional promoter for the early and late genes. My favorite gene is going to be the late genes because these are the late coding regions for the viral capsid proteins and the agno protein. Makes that nice icosahedral structure. That's the VP1. And this is the protein that engages with the cellular receptors that I showed you in a previous slide. And then the agno protein, that's responsible for the assembly of the viral capsules and the release of the virion from the cells as an infection progresses. All right, so the BK viral life cycle, I thought this was worth talking about for grand rounds. Okay, so VP1 mediates adhesion and viral entry into the host cell via these B series ganglioside receptors, GT1B, GD1B. We pretty much know these are involved. Some other molecules may be involved as well. BK, once inside the cell, it's transported along microtubules and it traffics to the endoplasmic reticulum. And that takes between 18 and 16 hours post-infection. Okay, the virus then undergoes partial capsid uncoating within the ER and gains entry into the cytosol. Hasn't entered the nucleus yet. So it's imported into the nucleus via binding of the VP1, viral protein 1 and viral protein 2 to a nuclear receptor yet to be identified. And then the BK virus begins early gene expression by about 24 hours post-infection. Expression of the large tumor antigen facilitates the assembly of replication complex, and DNA replication follows using the host machinery. Finally, the late genes are expressed, the capsid protein is expressed, virions are formed within the nucleus, and the virus is released via unknown mechanisms at this time. All right, let's go from the microscopic world back to the macroscopic world. And I think that this is important because we saw that history that we use a lot of different language. I've noticed when we talk about BK, there's BK viremia, BK DNAemia, and I think that it's very important that we kind of use the same language when we talk about this in clinical practice. So this is from the 2024 International Consensus Update Guidelines for BK. The only three that I really wanted to make mention of was probable, presumptive, and biopsy-proven BK polyomavirus nephropathy. Probable is when the viral count is greater than 1,000 and less than 10,000 copies per cc in plasma. Presumptive BK virus nephropathy is when it is greater than 10,000. And then certainly biopsy-proven BK virus nephropathy, well, that's when you have proven polyomavirus nephropathy, right? SV40, different polyomavirus stain. So we prove that's there, but then you also have a specific serum marker. Okay, then we can make the diagnosis definitively. Epidemiology and natural history. This is an opportunistic pathogen. Initial infection is via, we think, the respiratory or oral route. 80 to 90% of the general population is seropositive by 4 years of age. And then among that seropositive population, about 10% of us will shed the virus periodically in our urine, okay? But this is that when you talk about the natural history of BK, this is what gets frustrating, right? Because when we have the pyramid, right, we talked about a few weeks ago, we don't have the meta-analyses, the systemic reviews, and really the randomized controlled trials. It's prospective cohorts, retrospective cohorts, and case series when we talk about BK natural history and BK management. Challenging, right? We have gaps in knowledge. Questions we want to ask ourselves. What % of recipients develop BK polyomavirus viruria? Of those, what get DNAemia? What % of those develop BK virus nephropathy in the modern era? When nephropathy is present, what is the incidence of graft loss in the modern era with current disparate management among transplant centers? What is the rate of BK clearance with disparate management among transplant centers? So the natural history, just holistically, right? BK viruria precedes BK viremia, precedes BK virus nephropathy. So asymptomatic BK virus viruria, that's going to be seen in about 30% of kidney transplant recipients. That's when we can start to see shedding of cells of the urinary tract, such as from the ureter into the urine. And that's a characteristic cell there. You see the high nuclear to plasma ratio. Maybe you see some nuclear inclusions. And it's hard to see, but another characteristic is a dense ring of chromatin around the nucleus. So after the onset of viruria, nearly 50% of those recipients develop viremia during the period of 2 to 6 weeks after transplant. So that's about 15% of kidney transplant recipients. Okay. So plasma BK viral loads greater than 10,000, that's associated with higher rates of BK virus proven nephropathy. Loads greater than 1 million are predictive of extensive BK virus nephropathy. All right, so this is me being frustrated by the natural history of BK virus in the modern era. I don't want to be that guy. I want to be that guy, top right. So I managed to find a recent study that I really liked, and I hope you like it too, that I want to go through with you, and it's all about the natural history of BK. So this was Transplant Infectious Disease 2024, prospective observational study across 6 U.S. transplant centers. University of Kentucky was one, UAB was one, and the objective was to define the natural history of BK infection in a realistic modern post-transplant cohort, including incidence of viremia, reduction in GFR, identify risk factors for BK virus reactivation and BK virus associated nephropathy. So we had 335 kidney transplant recipients enrolled, and they were monitored every 4 weeks for up to 24 months post-transplant. Immunosuppression protocols, induction and maintenance, and management of BK viremia, they were all done by the standard of the care at the transplant centers, okay? Outcomes are listed there: incidence of BK viremia, timing of onset of viremia, duration of viremia, incidence of biopsy-proven BK nephropathy, risk factors between donor and recipient, and maybe transplantation factors, frequency of graft failure, etc. Okay. So for those participants that developed BK viremia and they moved into the intensive phase where BK PCR was performed every 2 weeks, yeah, those were, so if you develop BK viremia, that's my point. You moved into the intensive phase of the study where you were then tested every 2 weeks. If you did not develop BK viremia, you moved to, if you had BK viremia and then you cleared it, then you were no longer in the intensive phase and you were in the post-viremia phase, where the frequency of monitoring went from 2 weeks to 4 weeks. So most participants received ATG, followed by FK, mycophenolate, and prednisone. Here's just a demographic table of the participants across the, the 6 centers. Pretty standard. Looks like us.
Do you mind? So the clear versus not clear.
Yeah.
They're with treatment, whatever they used.
Whatever. Okay. Kitchen sink, nothing. Yep. Whatever that center's using. That's why I like it. This is what they get. Yeah, it's realistic, right? So these are all the medicines, that various recipients received. I mean, look at that list. It's crazy, right? Go down, and then on the right, the antiviral drugs specifically that various participants received. And the ones in bold are the ones that there's literature to support their use in BK viremia specifically. And later on, we'll talk about IVIG and Leflunomide. Okay, so the estimated incidence of BK viremia was 17% for the cohort. So for the subgroup that cleared, BK viremia, they had a lower mean peak viral load compared to the subgroup with persistent BK viremia, kind of as expected. And that's going to be 85,000 copies versus a high of 776 thousand copies. And that, reached statistical significance there. Sure, I didn't skip a slide. There we go. Okay. So risk factors for BK viremia, using logistic regression, authors found that what I have highlighted there, those are the statistically significant risk factors. Male donor sex was associated with lower odds relative to female donor sex for BK viremia. And then self-reported recipient Black or African American race relative to white race was associated with increased odds of BK viremia. HLA-C7, right? That's on there. I don't know if there's going to get questions about that, but several observational studies have suggested that HLA-C7 may be associated with an increased risk of BK viremia or BKVAN. Okay. This is just showing the incidence of BK viremia versus months since transplant. And yeah, what's important to note here really on this graph is where is the slope steepest? Right, that's what I clue in on, and it's really between month 1 and month 4 to 5 after transplant. That is where the slope for this cohort is most steep in terms of incident infection. What about time to BK viremia and clinical and demographic covariates? Again, I have the one of statistical significance highlighted, and that's basically saying that those that identified as Black versus those that identify as white, their time to BK viremia was sooner to a statistically significant degree.
Andrew, I will do like questions now at the end. Do you know if this is adjusted for other things? Like, why would one race have more risk unless you're just like, did they receive more HIMO? Like,
it's not adjusted.
It's not adjusted.
It's not adjusted.
All right.
No, it's Cox proportional, so my assumption is it is adjusted.
I didn't think that it was.
Univariate or multivariate Cox.
Say one more time.
You can do either uni or multivariate Cox.
Right, but I mean, usually when people put all of this together, like the general discipline is that if you are going to put all of them together and use it a Cox proportional, you've used it in a multivariate fashion.
I don't know. I don't know.
Okay.
I'm not sure. I'd have to go back and maybe look at the supplement or something. What about the total duration of BK viremia? So the median clearance time for the entire cohort was 5.1 months. And what they saw was that the female sex versus male sex had a statistically significant more rapid clearance. Okay. And then what about BK viremia and changes in kidney function? So participants that remained in the intensive phase relative to the screening phase or those that cleared, and recipient female sex relative to recipient male sex, those were associated with significant declines in the estimated GFR. Female sex versus male sex was associated specifically with a 2.1 decline in eGFR compared to male. And then remaining in the intensive phase was associated with a 3.8 decline in eGFR compared to a 5.0 improvement in the eGFR for those that transitioned from the intensive phase to the post-viremia phase after BK remitted. For the incidence of BK virus-associated nephropathy, nothing's highlighted there, and that's because they found that there were no donor, recipient, or transplant history characteristics that significantly associated with biopsy-proven BK virus-associated nephropathy. Okay, so just to wrap this up, a few points from the study. The incidence of BK viremia and biopsy-proven or presumed BKVAN were 17 and 10%. Risk factors for BK viremia were donor female sex and recipient self-reported Black race. Recipient female sex was associated with more rapid clearance of BK viremia compared to recipient male sex. Both persistent BK viremia and biopsy-proven BK virus nephropathy were associated with more rapid decline in EGFR, for up to 24 months post-transplant. Whereas those that entered the post-viremia phase, they experienced a relative improvement in EGFR. So I read this study, I tried my best to digest it, and I was feeling pretty good. And then I went to the IST 2024 guidelines and I looked at their risk factors. And I'm like, well, shoot, hardly any of them are listed there. Older recipient age, BK polyomavirus seronegative status, right? Tacrolimus versus cyclosporin use, corticosteroid use, you know, the list goes on. So I returned to my frustration. And that's part of my point here when it comes to the natural history of BK. All right. So let's look at, you know, kind of shift gears here and let's look at nephrology and the pathology specifically, but it's gonna have an entirely clinical application. There are no tissue slides in this entire presentation. All right. So prior to 2018, that's when the BAMP guidelines were updated to really include BK classifications. So the pathological classifications of BK nephropathy, they really were inadequate as self-admitted by the authors of the update. And so, well, the question they wanted to answer back in 18 was, can pathological phenotype criteria in proven BK polyomavirus associated nephropathy, can that be used to predict clinical outcomes, right? So patient presentation at time of index biopsy, renal function over 24 months of follow-up, and can we use the pathology to predict graft loss? So they collected data from 192 patients. They correlated that with morphological findings from index biopsies in the past, and that revealed two independent histological variables to be most significantly associated with clinical presentation. Those are the PVL and the CI, which is the BAMP interstitial fibrosis score and the intrarenal polyomavirus load score. So that resulted in class 1, 2, and 3 polyomavirus nephropathy. I put a red flag there, and this is the red flag that the authors brought up, is that they were looking at kidney transplant recipients that received kidneys between 1996 and 2008. That's a different era, right? And so they knew that, and I'll address that in a second. But before they do that, let's just look at the classes real quick. Score 1, score 2, score 3 for the PVL. That's a percentage of positive tubules and ducts that have evidence of polyomavirus replication. You add that to the interstitial fibrosis score, and then you'll get the polyomavirus nephropathy class. So here we go. We're going from a red flag to a green flag. This was just published in 2018. Fast forward 3 or 4 years, and a lot of the same authors of the update, they went back and they said, hey, we want to look at transplants that are after 2008. So let's do the same thing. Let's look at transplants from 2009 onward. And they published a multi-center validation study in the modern era for the 2018 update. Retrospective multi-center cohort, all with biopsy-proven polyomavirus nephropathy and transplant after 2009. So they had 99 participants. Index biopsies, they were all centrally reviewed for BAMP criteria, and they were assigned a PVN class. No protocolized follow-up biopsies. Data on renal function, such as creatinine, was collected at months 1, 3, 12, and 24. And then at that same frequency interval, BK plasma PCR was collected. Treatment, immunosuppression protocols, et cetera, that was all local management. And then the outcome, of course, is can pathological phenotype criteria and proven BKVAN be used to predict clinical outcomes with this new, more modern cohort of patients. So baseline allograft function did not differ significantly between the 3 PVN classes. Timing of PVN diagnosis was strongly correlated with PVN class. Basically, the higher the class, then the later the PVN diagnosis. And then at the time of index biopsy, acute allograft dysfunction was most pronounced in class 3 compared to class 2, class 1, right? As measured by percent change in serum creatinine baseline to peak. Makes sense. At the time of index biopsy, plasma BK virus PCR readings were highest in class 3 compared to class 2 and class 1. Okay, so we're making a little sense here. So when on the left-hand side is serum creatinine as a function of time since transplantation, and each color represents a different PVN class. And on the left is the same thing, but with GFR between time of transplant and 24 months. In graphical representation. So PVN class versus graft failure, looking at, so all patients right there on the left, and then patients without other complications post-index biopsy, those complications being rejection, infection, recurrent de novo disease, or malignancy. And kind of what you can see that there were differences in graft failure rates among disease classes. So 1, 2, and 3, they're already apparent 24 months post-index biopsy, but not significant. Okay, so that's here. Then we expand that out to longer follow-up. So that's specifically graft failure was recorded for 63 patients who were followed longer than the minimum 24 months. And we start to reach a clinical significance. Graft loss is very uncommon in class 1, right? Even in patients with these other complications. So what about polyomavirus nephropathy class versus polyomavirus nephropathy resolution over a 24-month follow-up? So resolution, yes or no? There was a trend toward higher recurrence of disease in class 1 versus 2 and 3. Now, this only reached statistical significance in subgroup analysis evaluating disease resolution based on a negative repeat biopsy. And then what about time of resolution? That's dependent upon the means of assessment, biopsy and or BK viremia. Median time to PVN resolution occurred between 28 weeks there in class 1, and then 53 weeks in class 3. And the impact of polyomavirus associated nephropathy resolution or persistence on graft failure. Okay, so what we have here is we have serum creatinine versus a sub-analysis of patients of various polyomavirus nephropathy classes. And you can see, right, some of them, this class is class 1, no clearance, versus class 1 that had virus clearance during a 24-month follow-up period. Significant differences they're seen in class 2. Clearance also had a positive impact on function and disease in class 1, although that is to a lesser degree. Class 1 is the green right here between these two guys. And then clearance was less common in class 3. And that's just a single line. Okay. So you want to get the QR code again real quick? I'll talk a lot about the natural history of BK virus and kind of sum things up. I really like this graphic that I found from the new guidelines. So right here is just time of transplant, and then as time goes forward, you first get the BK virus DNA urea, BK virus in the urine, then decoy cells appear, and then you have DNAemia. There's a specific antibody level response. There's a T cell response, right? And it's about this time that we recognize the disease. Net state of immunosuppression goes down. We have our bump in creatinine, and now immunosuppression is still down. We're worried about rejection. Creatinine goes up, boom, rejection, and then net immunosuppression goes up. It's a story. All right, so all that just to say, this is my summation. Screen all adult recipients monthly until 9 months, every 3 months until 2 years. That is my summary of everything that we just talked about. All right, catch it early, follow the guidelines. Don't test the urine, that's not supported. Someone asked me that question. So let's talk about management. All right, so reduction in immunosuppression. That's the mainstay of management. All right, there are variations in induction and maintenance immunosuppression regimens. This makes it tough, right? We're rolling the boulder up the hill. We're frustrated. Immunosuppression reduction strategies are different. Target drug levels are different between centers, et cetera, et cetera, et cetera. And this makes it all very difficult to interpret the studies and then create guidelines. But so let me just give you an example of a study. So this is from 2012, a retrospective single center cohort with predefined standard operating procedure. That's an example of reduction in immunosuppression, right? So 65 kidney transplant recipients, they all had BK virus DNAemia. This is what they did. Step one, they reduced they had 25% simultaneous reduction of mycophenolate and tacrolimus dosing without any antiviral therapy. Then they moved to step two. If that didn't work, go down again, 25%, right? See if the BK level comes down. Go from there. And then they had a tacrolimus target, 4 to 6. Okay. And then then we come up with some outcomes that get reported. We report episodes of cellular rejection, right? Decline in GFR, how long the follow-up was, peak DNAemia, right? Time to resolution, et cetera, et cetera, et cetera. Right. But then you run into these criticisms, right? The reduction in DNAemia occurred at an average of 200 days post immunosuppression reduction, when other effects, right, such as time since induction immunosuppression might be confounding factors. These are the type of criticisms that we run into. Other problems. Several studies have reported no impact of immunosuppression reduction on graft survival. Yes, that is true, right? These may be hampered by shorter follow-up periods. That's also true. Graft half-lives are increasing, right? Sometimes they exceed 10 years in the modern era. That's also true. So we run into all of these issues. So now we have to navigate this, right? So navigating the choice of immunosuppression reduction. You could first reduce the antiproliferative. You could first reduce the calcineurin inhibitor. You could do them both simultaneously. Ultimately, you could do complete cessation of the antiproliferative if the BK-DNAemia doesn't clear. These are different strategies that we take. And we have evidence to support each. So this is an example of an MMF reduction for a strategy back from 2005. Single center, open label, prospective randomized trial. So de novo kidney transplant recipients with ATG induction. Some are on FK, right? Some are on cyclosporin. Most of them received azathioprine or mycophenolate. Well, they all did. BK virus DNAemia and it in the urine were collected weekly, for that period of time. And the treatment was identification of DNAemia, right, performed retrospectively that triggered discontinuation of the antimetabolite, right? MMF reduction first. If viremia failed to clear within 4 weeks, then the CNI dose was tapered to these trough cyclosporin and these trough tacrolimus levels. Okay, and then they report various outcomes. And I don't really want to go into the details here because it's not really my point. Because they're all going to report, right, how many developed viruria, how many developed DNAemia, what was the associated clearance, how many developed graft loss, and then what was the rate of acute rejection. So my point basically is that it's hard to navigate, right? But you have studies like this that support if your center wants to go the route of anti-metabolite reduction first in their how they combat BK virus disease. For the sake of time, I have other studies that I can go through. I'm going to skip that and go on to the next approach and maybe talk about a study or two. That's CNI reduction first and simultaneous reduction. So the first approach basically is to reduce the CNI by 25 to 50% in 1 or 2 steps, followed by a 50% reduction in the anti-metabolite if it doesn't clear, and then a subsequent discontinuation of the anti-proliferative drug. Or you can do a simultaneous reduction of both the CNI and the anti-metabolite at the same time. So I will go through this one. I liked this one. This is from 2019, a retrospective single center cohort with predefined standard operating procedures for screening and treatment. Many kidney transplant recipients, right? So BK virus DNAemia, i.e. viremic, was defined as that, having greater than 300 copies per cc. And then they defined probable, presumptive, and then biopsy-proven disease. This is what they did. Immunosuppression reduction. There were no other interventions. Patients with known BK virus DNAemia. Number 1 is the tract trough levels were set 1 step lower than intended by the protocol, than the typical protocol at that center, and targeted as predefined for the next period. So, for example, if it was 9 to 11, right, they would go down to 8 to 10. Okay. If BK virus did not decrease within 4 weeks, TAG trough levels were further reduced by another step as predefined for the next study period. And then step 3, if another 4 weeks goes by, BK viruses levels still don't decrease, then the dosing of the anti-metabolite was reduced in steps of 50% until discontinued. So outcomes: clearance after tacrolimus reduction alone for 39% of all patients, but response differed according to the diagnosis. Okay, patients with probable BK virus polyneuropathy, they cleared the disease in 63% by TAC reduction alone, and then another 25% required anti-metabolite reduction as that. So they went into that third step. Patients with more disease burden, the presumptive group, they cleared DNAemia by TAC reduction alone by 51%, and then another 48% needed to hit that third step and reduce the anti-metabolite. And then finally, with the patients that had biopsy-proven BK virus disease, that percentage that cleared with TAC reduction alone, that was only 21%, and another 61% required anti-metabolite reduction. So at 6 years post-transplant, other outcomes, no differences were seen among the 3 groups regarding graft survival. No, I got cut off there, I'm sorry. Probably, I think it was clinical outcome. But among these 3 groups, probable, presumptive, and biopsy proven. So this is just to say that there really is no correct approach, and there's really no wrong approach, right? You have to kind of take everything into consideration, maybe their immunological risk profile, maybe their BK viral load, how well they're doing with their medications, how frequently they're getting labs, things like that. And then, so you take the patient, you look at the data, you look at the latest guidelines, and then you kind of adjust their management accordingly. And then you hope that they keep getting their blood drawn so that you can follow the BK virus load. That's really, I think, that's kind of the crux of the matter. Last thing, right? Adjunctive treatments. I really wanted to get to this, talk about IVIG and leflunomide. So IVIG, some of the data. It's commercially available IVIG. That it contains a high neutralizing antibody titers against BK polyomarvirus. Okay, you can find neutralizing antibody against the genotypes, which I mentioned in the beginning presentation, 1, 2, 3, and 4. There are subtypes of the genotypes. I didn't get into that. There are only a few studies, however, reporting a trend of improved clearance of DNAemia in patients receiving adjunctive IVIG compared with historical controls treated solely with immunosuppression reduction. Okay, this is one of those studies. They looked at the effectiveness of IVIG in a retrospective single center cohort against standard of care controls. So both groups underwent reduced immunosuppression. Okay, conversion of tacrolimus to cyclosporin. Some centers do that. And mycophenolate to leflunomide. Some centers do that. They received oral ciprofloxacin, and then IV cidofovir was given to some. Okay, so this is a lot of treatment, a lot of management. They had biopsy proven BKVAN occurred in 50 kidneys at 7 months after transplantation, and then this was prior to 2018, so they called it histological stage B, right, prior to the BVN classes. They gave the IVIG at this dosing, and then the IVIG group, quote unquote, more effectively cured viremia and BK immunohistochemistry from repeated tissue sampling, and resulted in faster and more complete resolution of viremia. There were fewer graft losses that occurred in the IVIG group. Okay, so this is just one of the example of one of the few studies that support the use of IVIG. And then I think lastly, just leflunomide to end up here. All right, what do we know? It's an anti-proliferative immunosuppressant. The active metabolite is teriflunomide, or LEFA. That inhibits T and B cell proliferation. And interestingly, other derivatives of leflunomide, like specifically the FK778, they were investigated as immunosuppressants in kidney transplant a couple decades ago. Didn't quite make it to the market, from what I understand. All right. So we got this study here, and the purpose of this study was to investigate the antiviral effects of the active metabolite leflunomide on BK virus replication in human renal tubular epithelial cells. Interesting finding. The compound did inhibit BK virus genome replication. So late protein expression, that would be the VP1, VP2, and VP3, and then virion assembly and release. So disrupting the nuclear replication architecture. This reduced extracellular viral load by approximately 90%, but it came at profound renal tubular epithelial cell anti-proliferative effect. Right, so that was the downside, and then the clinical data is just conflicting when it comes to leflunomide. Here's a study though that is clinical from 2007, prospective single center case series, kidney transplant recipients with biopsy proven BK virus nephropathy. Their treatment was immunosuppression minimization. And then mycophenolate was switched to leflunomide. BK-DNAemia cleared in 42%. Serum creatinine concentration stabilized or improved in 66%. They dosed the active metabolite to that concentration. And then graft loss occurred in 17% of patients, just a case series. Okay. Different study, same number of participants, and same treatment. This was a prospective multi-center case series with predefined standard operating procedure for treatment. BK virus cleared in 12, that's 92%, with stabilization of serum creatinine. Drug levels were monitored and dosed to a target concentration of about 40, but there was no correlation between drug concentration and treatment response. And then just 1% effect experienced some adverse effects, probably attributed to the leflunomide pain and paresthesia. And 1 patient had graft loss. Okay, so this is what the new 2024 guidelines suggest when it comes to adjunctive treatment. Only IVIG, right? Only IVIG made the we suggest. Okay, that's it, right? All right. So it's murky. Long story short, it's murky. But my favorite part of this entire talk when I was putting it together was honestly the beginning. I think that the research that the BK researchers are doing around the world in terms of making more clear the virus structure, how it interacts with the cell surface receptors, right? Running that lock and key through molecular libraries to see if something works, maybe disrupt the entire disease process. That's really cool. So we'll probably have advances before I retire. So we'll stay happy about it. Not too pressure.
I have a question.
Yes.
So have you encountered any studies on the use or substitution of mTOR in lieu of CNI?
Yeah, there are. Yeah, there are studies on that. I think there are cohort studies and both were positive. I can't remember exactly, but I want to say the gist is that both were positive in terms of favorable clearance with mTOR versus CNI, but the data is still not robust enough to make it into, for example, the 2024 guidelines. But they do make mention of that. And Belatacept is mentioned as well. They may comment on that.
It's just that when you're pushed to the point of desperation, there's nothing else to do, and you can control it. We've done that before. We stopped CNI and just hope and report. Replace the patient on virology.
Yes.
And it disappeared. So, and there are literature on the supposed antiviral association of mTOR. That's why.
I was just looking.
You've heard of these cytotoxic lymphocytes off the shelf that people have used for certain other viral diseases. I think they do that in Cincinnati. So we actually had a patient who lost his first native kidney. He had a bone marrow transplant for Fanconi type anemia and got a transplant from his mother, and then he got a bone marrow transplant from a mother, and then he went on and got kidney failure and from BK virus because he was so immunosuppressed from the bone marrow transplant.
Yeah.
It's not a kidney, but then the BK knocked out his native kidneys, so they failed and he needed a kidney transplant. And they tested mom's T cells and she had 0 BK virus in her urine, and he had lost his natives from BK. So we tested her T cells to see if they would kill the BKs, and they did.
They did.
So we were able to transplant mom's kidneys into this kiddo, and with no transplant immunosuppression and no specific things. But then later we've this next step, the people like in Cincinnati and other places develop specific cytotoxic T cells that kill VK virus or ABV or CMV, and you can they're starting to develop ways to give these T cells to kill them. But the trick is with the transplant, we've probably knock out the transplant too. So for a non-transplant, non-transplant problems.
Yeah, actually, there was a series of clinical trial. Actually, they did at MD Anderson. They have 7 patients. They used T cell therapy, which is commercially available for the clinical trial. And what they saw is viral load was better, but GFR was not better.
GFR doesn't help.
So they are optimistic for further outcomes.
Yeah.
Absolutely. Well, Dr. Slater, beautiful presentation, first of all. And I have to say, I think one thing that COVID and COVID research taught us is how short the timeline can really be, right? To have a virus, to identify its structure, identify what prevents it, right? The vaccine and the medication to treat it.
Yeah,
It's often done on the scale of months, maybe to,
that's
years.
true. That's
So,
true.
so I don't think you need to retire for that. We just
Yeah.
want funds and
There's just so
interest.
few people, like
Yes.
you read, you read like, I probably read 5 to 10 papers just on BK virus structure, and the authors are all the same, just in different order. Can do a second this time.
Yeah. Technology is there. That's the thing. The machinery is there. It's just a matter of funding it.
Yeah.
Oh, yeah, it's now the prime time to get funding for everything.
Sure.
Very good.
Thank you.
Thank you.
Oh, yeah.
Yes.
Yeah. I didn't take a deep breath.
I'm sorry.
All this schedule has everything, so I'm just trying to give myself grace
She's
with
the
like
one
the time that I
let me
have
make sure I
to do
was
it,
next to you.
thinking that it's.
Yeah.
But I know that
Oh,
it's
You cannot.
I wish I would have had the mental power to analyze it
I
better.
think, I think
It's
this
just-
one logical.
I'm already
Because you were hoping that will work, but
me a
now
while
because you
to
don't
like
expect
get through
your diagnosis is.
journals, and it's something that I'm talking about.
It's like, it's like,
Get the line and I'll do the answers for it.